let's talk about the study that just recently put out. So right now it's in a preprint form, but so there's a ton of data there. In this research, you looked at a Jamaican lion cultivar and
Yes.
mapping.
That was. Oh, go ahead.
I was just to say you were mapping the parents, the siblings, and you were looking at genes related to cannabinoid expression, sex expression, and pathogen resistance. Is that right?
Yes, yeah. And so just to, caution all of it, it is a preprint. It's not been through peer review, but all the data is public. So anyone can grab the data and download it and play with it while it's going through peer review. Koji is a great place to go if you want to look at the data. It's a genome browser. Comparative. If you just Google COGE or comparative evolution or comparative genomics genome browser, you'll see a search in there for Jamaican lion or for cannabis, and you'll see a certified reference up there that has a lot of this data linked in. It's got great graphical tools, so you can kind of play around with this data yourself. But yeah, so the purpose of that project was to first build some good cannabis references. Back in 2011, we sequenced a ChemDog strain with these short read Illumina tools. Mm-hmm. And we probably did 1 billion reads. And when you try to assemble those reads, the genome is so complex that you only get like puzzle pieces out that are about 3,000 letters long. This is what we call a contig size in genomics. And you want those contig sizes to ultimately be the full length of a chromosome. Mm-hmm. It's rare they ever end up that way. They usually are broken up into a couple pieces because the centromeres are hard to get through and some of the telomeres are hard to get through. But the human genome has 1 or 2 chromosomes after 15 years of being declared finished or done, finally tip to tip. So we're going to do much better than that in cannabis. It's going to happen faster because we get to stand on their shoulders. But so there's this term in the genomics field that measures the contiguity of genomes, and it's known as the N50 number. This is the average size of the length of the pieces of your puzzle. And cannabis chromosomes are usually 60 to 100 megabases in size, and you'd love for your contigs to be perfect stretches of 60 million bases long. I think the largest one we have in one of ours is 40 megabases, so it's close. We're almost to perfection. But the references we had when we started this project, the N50 size, I think the largest one at that time in GenBank was about 150 kb, so 150,000 base. So a big improvement from 2011. And then John Page's group put a few out a few days after we released the Jamaican lion one. They put one out from Purple Kush and Finola, and those inched up toward 400. I think Sunrise put one out that's in the 750 range, 750,000 bases. But the real assembly gurus don't get excited until you're over a megabase. That's kind of like a mental threshold that they want to see before they start considering a reference grade genome. And we still weren't quite there yet. So we decided to keep sequencing, and all that we did different than the people behind us was that we just jumped on board of the newer sequencers. The latest and greatest chemistry just pushed the Jamaican lion reference above the megabase mark. So it's currently at around 3.2 megabases on the preprint that we put forward. So the average stretch of sequence in there is a stretch of 3.5 megabases long. Now, the other thing that's important is looking at the quality of the sequence to make sure there aren't a lot of errors. Because when you have 58 million variants, you don't want to be spending your time on 5 million ones that are wrong. It's a lot of error to sift through. So the more later sequencers tended to have a lot better error rates and more accurate data. Now, the other thing that you tend to do to really build a strong reference genome is you like to sequence a trio, which is a cross, basically, a mother, father, and an offspring. And the reason you do that is you can then triage your errors that don't make any Mendelian sense from the ones that should. We call these Mendel errors, and there's less than like a 0.2% Mendel error rate in the Jamaican lion trio that we have. So what that means is you look for variants in your offspring, and you say, do I see evidence of this in either of the parent? If it's not in either parent, it's probably a sequencing error. And you use that information to basically categorize what variants are real and what are wrong. The other thing that usually has to go on when you're dealing with a new species, and we're doing some work with this on Google and a few other parties in the space, is you usually have to retrain your algorithms that look for the variants when you're dealing with a genome that's as polymorphic as cannabis. All these tools were trained on humans, so they're only expecting a variant every 1,000 letters. When they start seeing one every 50 bases, they start throwing everything out, saying this is a different organism, get the hell out of here. So you have to go back and retrain all the base callers and all the variant callers to start calling the variants more appropriately. And they usually take the tools that are used for mosquitoes. Mosquitoes have a variant like every 20 bases. They're one of the most polymorphic species out there. And so the tools that they did on Gambi, they can then, Anopheles Gambi, they can then kind of tune to be somewhere between humans and Anopheles and settle in on cannabis. So we went through the effort of doing that type of training with the tools that we were using, so we get the base calls really accurate and the assembly is very accurate. And then once you have that sort of trio, that reference set, you can then confidently take Illumina data and map it across all of those references and start asking questions like, are there entire segments of the genome missing? What's the polymorphism rate in all the genes that are out there? How many highly damaging variants do we have in a given cultivar? In other words, mutations that actually destroy genes. Right. How many of them? There's like over 2,000 of those in a given genome. There's that many genes that get obliterated through polymorphisms. Once you also have these references, the next thing you want to do to curate them is you go and sequence the RNA off of as many tissues as you can. And what that does is it tells you what genes are being turned on and where they are in the genome. Today, we don't have great tools that can just scan a reference genome and say, I can see where all the genes are just algorithmically. They really want to see biological evidence of how those RNAs are getting written off the genome. So if the listeners aren't familiar with DNA and RNA, the analogy you often hear is that DNA is like information on a hard drive. And the RNA is what's in your task manager, which programs are actually getting executed off that hard drive. Not every cell in the genome or in the plant is going to be expressing all genes all the time. Right. A trichome is going to be expressing a lot of THC synthase, but it may not be expressing any of the root proteins that are needed to interact with the mycorrhizal. So you do this to try and get as many pictures of what genes are on and which tissues. This helps you annotate the genome so you know where all the genes are. And I don't think we're done with that process yet. We did this across 5 tissues. We found about 27,000 genes in the female genome and about 32,000 genes in the male genome. And those numbers are likely to increase over time as people survey more tissues and do more sequencing. One thing that is really a bit of a breakthrough that's happened this year in the sequencing platforms is those same single molecule sequencers can now be deployed on RNA molecules. And in the past, we didn't really have that. We had these short read sequencers trying to ascertain where the RNA is mapping to the genome. And that's really hard to do because RNA is kind of like a fragmented hard drive. It never reads a gene from a capital letter to a period as a perfect sentence. What tends to happen in the genome is your gene gets scattered across the genome in like 10 different pieces, almost like fragmented data on a hard drive. And there are tools in the cell that when you make that RNA transcript, they go and splice out the regions that are the exons and make what's known as a transcript. And the process of excising those exons, it can do in a combinatorial way. So you can have any given gene through a process known as alternative splicing of that RNA can make 3 to 10 different proteins. So the protein complexity is not a one-to-one correlation with the genes that are in the genome. But having these sequencing reads that can read across the entire gene's RNA in one shot means you can accurately place all of those exons back into the genome. Wow. We haven't had that in cannabis until this year, actually. I think we were the first people to probably put out any PacBio RNA isoseq data that gave us really clean information on intron and exon boundaries. That's something that's really hard to do with short reads, particularly in very polymorphic species. It's a bit of a challenge. So we now have that backbone inside that Koji browser. We can see where all the transcripts are in 5 different tissues. And then we also started to play around with methylomes. This is the epigenetic code, and it's never been done in cannabis before. It's very hard to do in cannabis because cannabis, the genome is 63% AT. So there's 4 bases in most genomes, A, T, C, and G. And the human genome has roughly an equal amount of all 4 letters, making it a fairly easier genome to work with. But as the genomes skew themselves more in the direction of one nucleotide pair or the other, it's kind of like putting a jigsaw puzzle together where all the pieces are the same color. You end up having less entropy and signature in each of the pieces, and so you find yourself confused more often. Well, when you want to do methylation analysis, you have two choices. You can use some of these single molecule sequencers, but they're not good at doing it in plants because plants' methylation patterns are far more complicated than microbial ones. So you can hit microbial and maybe human methylation patterns with the single molecule sequencers today, but they don't handle plants very well yet. So we had to resort to a tool that's known as bisulfite sequencing. This is something that will convert the DNA if it's not methylated into it. All the cytosines in the genome, if they're not methylated, they get converted into a T, into a different base. And so by sequencing this before and after a conversion like this, you can figure out all the cytosine bases in the genome that are protected by the methylation mark. However, the genome goes into something like 88% AT. It becomes so AT rich that you can really you get lost trying to put the reads back into the genome. You can only map half of the genome back with this tool. But it's the only thing we've got today to handle methylation. And so we ran that on 4 different tissues to get an understanding of where all the promoters are in the genome. The promoters are the regions in front of the genes that are usually responsible for turning the gene on or off.
Right.
And when they're methylated, they're silencing the gene. And when they're unmethylated or hypomethylated, they're turning the gene on. And so we've got beautiful pictures now of THC synthase and the adestin gene, which is involved in some seed development, those methylation patterns getting turned on and off on DNA. One reason why we're excited about this is it does give you it gives you better paleogenomic tools. If you ever want to look at ancient cannabis samples, you can't do it with RNA. RNA is one of these molecules that breaks down really readily and very quickly. Right. It's that way on purpose because it needs to make those RNAs to send a signal and it needs to kill those programs afterwards. And so there's all this feedback mechanisms in cells to destroy RNA after it's been expressed. Methylation, however, sticks around for a really long time. You can actually pick up methylation signals on ancient DNA samples. So it can give you some understanding of what genes are turned on and off across long time horizons. And so there's an interesting aspect to methylation data in that regard. But that information is now together and public, and people can kind of comb through it. That was leading to your final question, which is, what did we see in this data regarding pathogen response?
Right.
And on the pathogen response front, we took a little bit of a myopic approach in that we looked at all the genes that have been published in hops that play a role in pathogen response, particularly powdery mildew resistance, because powdery mildew is a type of pathogen that's very cheap to detect. There's not only PCR assays for it, you can visually see it. So you can tell if a plant is powdery mildew resistant or not, if it's surrounded by other plants that are loaded with it and it seems to not have it. That's not the case with a lot of other pathogens. You can't see them, so it's hard to phenotype them. But we looked at chitinases. Chitinases are enzymes that are throughout the kingdom, the plant kingdom. You see them in all plants. And you also even see them in fungi.
That's what I was about to say, yeah.
Yeah, so it's an interesting game. Some fungi express these to kill other fungi. Trichoderma is one that makes chitinases to kill aspergillus. So it's a weapon that's used to fight off fungi because the cell coats in fungi are made of chitin and another molecule known as a beta-glucan. Yep. And beta-glucan is something plants have evolved to fight as well with proteins known as thaumatin-like proteins. These things chew the other thing that's inside of a fungal cell membrane. So we looked at those two gene classes and then another one known as MLO, which is a mildew locus that was discovered, or mildew resistance locus that was discovered in hops and in other plants as well. But there's some work done in hops trying to map this out. So all in all, that was about 82 genes that we just went through and said, are there any large copy number changes in these genes that might correlate with pathogen response? And we're starting to find some of those patterns in Jamaican lion. There's a handful of thaumatin-like proteins that have copy number amplifications that seem to correlate with powdery mildew resistance. And there's some chitinases as well that are sticking out. So those have been interesting to look at. But I wouldn't say it's the final chapter on that story because I think we have found the genes responsible for powdery mildew resistance in Jamaican lion, but I don't think you can necessarily apply those to all the other plants out there because I think there's 5 to 10 different modes of resistance out there, and we just happen to be looking through the lens of one of them.
Right.